Thank you all for coming. And first of all, we would like to start with the prayer from Basana.
Sambonani, Lumina, Kuyamoga, Shini, Kamara Mungosana. I'd like us to make a short prayer. Can we bow our heads and let us pray?
Can you please settle down? Thank you. Can you please get a real page to describe the subject
Welcome, ladies and gentlemen. Can I have everyone's attention? Man, I have everyone's attention. So I go by the name of Professor. I call myself an extraordinary man who creates extraordinary things. Welcome, extraordinary man who creates extraordinary things. So I'm going to talk about the structure of a DNA, the structure of an RNA. So let me start with a nucleic acid. So we know that we have the nucleic acid, and it has a nucleotide. This one is called the monomer, the building block of the nucleic acid. It makes up, this one makes up, it builds the building block of this one. It makes up the nucleic acid. So when we talk about the nucleotide, we have three types of, it consists of three types of things, which is the phosphate group, It consists of the nitrogenous base. And in DNA structure, there is a deoxyribose sugar. You know it, right? So it goes like this. This is P, you know, the rough sketch. Yeah. This is the deoxyribose, and this is the nitrogenous base. So in RNA, this is the DNA structure. The DNA structure. So in RNA, things are different. So it's P over here, then yeah, something like this. So it's P, R, and it's N. So in RNA, the structure of an RNA, here there is a ribose sugar. So deoxyribose is replaced by ribose sugar in RNA. You understand, guys? So I want us to understand, just we cannot live here without grabbing something. You know? We have to grab something. So what I'm going to talk about next is the DNA. So we know DNA has thymine, adenine, cytosine, and guanine. We know that when we describe the process of DNA, we find this fourth stuff. So we know that in the DNA process, we know that the double helix will start to unwind. When the double helix starts to unwind, what happens? The weak hydrogen bonds break. When the weak hydrogen bonds, what happens to it? They form two separate strands. Two separate strands, and one forms as a template. One forms as a template, and then they form a complementary DNA strand, which thymine pairs with adenine, cytosine pairs with guanine. Can we repeat that, guys? Thymine pairs with adenine, guanine pairs with cytosine. Can we repeat that? Come on, I'm not running a choir, guys. Please. Adenine pairs with adenine, thymine pairs with adenine, cytosine pairs with guanine. Please, man. So in RNA, so this is the DNA. This is what happens to the DNA. So in RNA, we have adenine. The complementary of the adenine will pair with uracil. So we are replacing this thymine here with uracil, and then cytosine will pair with guanine. So adenine will pair with uracil. Guanine will pair with cytosine. So what happens is that we know these two guys, Francis Crick and James Watson, these two Englishmen. These guys are crazy. How come are they coming up with something like this? Who can come up with this out of their mad minds? So I have thought in my mind that these two guys have used this formula of imagination. Imagination, I remember a quote from Einstein saying that imagination is powerful, it's more powerful than knowledge. He uses that to describe theories like the quantum physics and all that stuff. So, yeah, this, guys, is the process of, is the structure of DNA, and this one is the structure of the RNA. So, so here, guys, we see that here we have weak hydrogen bonds, which combines the adenine and the thymine together. Here we have the ribose sugar and the phosphate group. Do you understand, guys? Do you get me, guys? And then here we have uracil. Let me just make it quick. We have uracil with ribose with adenine here. So that previous one was the DNA structure. This one is for the RNA, where adenine pairs with uracil. This is the hydrogen bonds, the weak hydrogen bonds, and this one is the ribose sugar. This is the phosphate group. You understand, guys? So I'm gonna leave you with the quote wrote by Jim Quick. He said, "I believe these difficult times can define us. I believe these difficult times can diminish us. I believe these difficult times can make us into humans who explore the world, who help other people, and make the world a better place." Thank you, guys.
Okay, thank you, Raw Khalid. Can we please have row teams with DNA replication?
Good morning, everyone. Hi, can I please have your attention? Hi, can you please hear me now?
Yeah, guys, please let's give attention to the person who is discussing. If you give them attention, they'll give you also attention when you are describing. Yeah. Thank you very much.
So, good morning, all. My name is Tamina, and I'm currently representing Wilkins, Florida. So, I chose DNA replication. And DNA replication is said to be the process whereby DNA makes a copy of itself, so it makes a replica. And DNA replication takes place just before mitosis and meiosis, which is cell division. And this happens in interphase. So With all of this, we know from the person before, we know the structure of DNA and how it is formed, and it has a double helix. So the first phase of DNA replication is where the double helix unwinds. So the DNA strands unwind. The second place is where, from here, our weak hydrogen bonds break down, and this is where it starts to unzip and separate. Then from there, each of the DNA strand forms a template, and this template is a strand depending on complementary basis. So it has adenine and thymine, guanine, and cytosine. From there, nucleotides within the DNA strand join together to build up the base. And this is where it forms another DNA strand. And from there, this is where identical cells or identical molecules are formed, which is this part over here. So from here, the first step is it unwinds, then unzips, a template is then formed, complementary bases, well, we have complementary strands, so they on their own. And then finally, this is where we get our two molecules, our DNA molecules, where one is from the original and where the next is a newly formed DNA strand. Thank you.
Okay, thank you, Royal Kings. Is there any teacher or anyone who'd like to add anything to what she
Thank you, the MCs of tonight's program. Ladies and gentlemen, good morning. Good morning, everyone. Morning. Yeah. When we look at what we are doing today, guys, we are here both to learn, to unlearn, and to relearn. Okay? So it means you must pay attention to the person who comes in front here to discuss something. Why you may know something that the other person doesn't know, or you may know something which this person is going to debunk. Okay? I don't want to speak much about the structure of the DNA. I just want to add something small about that. That this person is supposed to basically describe how best we can describe the structure of the DNA. And then number two, as he said, we looked at the bond, the pairing of the nitrogenous bases. That was right. And basically, he was supposed to add as a bonus of where do we find the DNA. Okay, that will help us a lot. So I guess we all know where we can find the DNA, and we have three types. One, the nucleus, the mitochondria, and the chloroplast. And then about the DNA replication, I'm just interested in one thing. Where do the nitrogenous bases that come to form the template come from? These nitrogenous bases are free nitrogenous bases found within the nucleus. That must be made clear. Thank you very much.
Thank you so much.
Good morning, guys. Morning, morning. Can I get your attention, please, everybody? Thank you. The structure of the DNA and DNA replication, the people that came out, or the presenters rather, touched on our topic, which is transcription. So a lot of the things that we're going to say might sound redundant, but it's important. Okay, so we're going to start with, or basically, transcription. The key or the main focus in transcription is mRNA, right? So mRNA, the definition of mRNA is mRNA are molecules carrying the coding sequence for protein synthesis. So before I get to transcription, I want to touch on protein synthesis. So what is protein synthesis? My partner Kayla will give you the definition.
Protein synthesis is the process by which protein is made, like the cells producing new enzymes, hormones, and new structured cells.
So as I said, transcription is part of protein synthesis. And in protein synthesis, you get two parts. You get transcription and translation. But we're going to focus on transcription.
Thank you very much. Have a listen now.
College, can you guys please just calm down? Thank you. Okay, because of the interruption, we're kind of going to start from the beginning. So, as I mentioned earlier on, that transcription is a part of protein synthesis, right? And it's made up of mRNA, or the main focus is mRNA. And the definition of mRNA is mRNA are molecules which carry the coding sequence for protein synthesis. And my partner Galen will give you the definition of protein synthesis.
Protein synthesis is the process by which protein is made by each cell in the organism, making new enzymes hormones, and new structures of cells.
Okay, so in protein synthesis, there's two parts, it's transcription and translation, but we're going to focus on transcription, right? So in transcription, it goes like this. The process of transcription goes like this. We first have a double helix structure which unwinds, right? This is unwinded. And then the weak hydrogen bonds break and it unzips, basically. And then one strand is used as a template to form an mRNA strand. We got that, guys. And then from there, the mRNA strand uses free mRNA nucleotides from the nucleoplasm. Oh, sorry, I'm fumbling. It uses free mRNA nucleotides from the nucleoplasm. Then mRNA is complementary to the DNA. Galen?
mRNA is now ready. It now has code for protein synthesis.
Okay, the last point we're going to touch on is basically the movement of it. So, Kalen, just to be sure, where does it move from?
The nucleus,
And then where does it move through?
the nuclear form,
And where does it move to?
the cytoplasm,
And what does it attach to?
the ribosome.
Okay, so we got it. The mRNA moves from the nucleus through the nuclear pore to the cytoplasm and attaches to the ribosome. And that's our time. Thank you.
Would any teacher like to add on that?
Thank you very much, guys. Thank you very much, guys. Before I add something, I just want to pass this small announcement. They have told us that electricity is going in just 10 to 5 minutes. So, what does it mean? Does it mean that our life is gonna go to the end? I don't think so. We are planning to shift to the computer lab. It means that the space might not be enough, but we're gonna have to squeeze ourselves in there, so that because we have the generator there, the generator doesn't reach here, and then as we are transferring, we will be recording. Then we're gonna put it live, maybe after 20 minutes. You understand? So it means that even if you are discussing when the electricity is gone, still your discussion will be live. So it means that the live will be playing by some few minutes. Thank you very much. Another thing is you talked about transcription. I know that everybody here has a zip, isn't it? Zip up, zip down, zip up, zip down, zip up, zip down. You know zip, Yeah, sometimes some of you, these things you don't know them, yet you do them every single day. Yes, you cannot remove your gene if you have not zipped down, isn't it? DNA, double helix unwind, weak hydrogen bond break. When you zip down, you are breaking the weak hydrogen bond. That's why you say it unzips. Weak hydrogen bonds breaking is the same thing as saying then it unzips. You understand? So when you are marking DNA double helix, give you a tick. I think weak hydrogen bonds break will give you a tick. If you don't say weak hydrogen bonds break and you say it unzips, it's the same thing. Ne? We give you the same thing. Ne? You form two strands. You know, when you unzip, yes, you form two sides, which we call the strands. Here comes the question. These two strands, are they going to become templates? Both of them? No. Because the question can come and say, give the difference. Someone was describing DNA replication. Give a difference between transcription and DNA replication. You form two templates for DNA, sorry, for transcription. You form two templates for DNA replication, and you form one template for transcription. Is it clear? Okay. Another marking point. Now you form two strands, and then one strand is acting as a template. The word one must be there. We give you a tick. Is it clear? That's how we mark. If you don't have the keywords, even if you write thousands of words, we won't give you a tick. We're not about how many words you have done. No, we are about which kind of fact have you given. Is it clear? You're gonna be writing your tune, but make sure that you write facts. You don't stop there. Now you continue. You have formed one template is acting as, sorry, one strand is acting as a template. I'm gonna ask you a question. What is a template? You are saying one strand is acting as a template. What's a template? In layman's language, who can help me? Who can help me? You know, if you don't help me, I'm gonna just pick you up. Ne? Yes. Who can help me? Who can help me out? Template, what does it mean? Ladies, ne? You know me, I'm a guy, ne? I don't usually check myself. But ladies, every single day, you cannot even spend a full day without checking yourself, your face. You have to check and see if you go to the toilet. That's why in the ladies' toilets, you find mirrors there. They have to check all the time. What do you see in the mirror? What are you? You are the template to the mirror. You understand? You form a copy of yourself in the mirror. So that's exactly what DNA does. So when you talk about a template, that's how it works. Is it clear? But don't write a mirror in exam. You get 0. Then this template must have complementary what? Bases. Because we're forming messenger DNA. T is gonna go with U. Is it clear? Is it true? No. You are wrong. Now they're saying yes. Is it true? No. T is gonna go with you. Is it true? No. T is gonna go with exactly. And then A is gonna go with me, then. Oh, with you. All right. And then C is gonna go with what? GX. That's what we call complementary base pairs. But these base pairs, what do they use? They use free. We want to see this word in your writing. Free RNA nucleotides. Where are they found? In a leg? No. In the nucleoplasm. Free RNA nucleotide. Remember in the DNA replication, you said use free DNA nucleotide. Here we say using free RNA. If we don't see the word free RNA, 0. Free RNA nucleotide. So you form a complementary strand which is a messenger, and then now it moves. We are saying it moves out of the nucleus. Then it goes to the cytoplasm. Where exactly? Ribosome. Thank you very much, guys. Thank you very much.
Okay, guys, can you please welcome Skyline, which is the translation.
Hello, everybody. How are you doing this morning?
Fine.
My name. Hi. So forgive me for my English. I'm a little bit afraid of like how you understand. I'm going to describe. Uh, I'm going to describe the second stage of protein synthesis called translation. What is translation? Translation is a process whereby different amino acids are assembled to form a specific protein. This occurs in the cytoplasm in the organelle ribosome. There are three, which is called the tRNA in the. It happens in the